Hello students, welcome to EPG Pathshala. I am Suma Saad from Jamia Hamdard University, New Delhi, and the topic is In Vitro Studies, Paper Product Development Part 2. Now we are talking about the learning objectives. In this module, we will study about what is in vitro studies, advantages of in vitro studies, limitation of in vitro studies, various models for in vitro studies, and in the last, common product safety tests. In vitro studies are performed with microorganisms, cells, or biological molecule outside their normal biological context, test tubes. These studies in biology and its subdisciplines are traditionally done in lab where such as test tube, flask, petri dish and micro-titer plates. Studies conducted using components of an organism that have been isolated from their usual biological surrounding permit a more detailed or more convenient analysis than can be done with whole organism. However, results obtained from in vitro experiments may not fully or accurately predict the effects on a whole organism. In contrast, in vitro experiments in vivo studies are those conducted in animal, including humans and whole plants. In vitro studies consist in subjecting cells or tissue to low frequency electric and magnetic fields. The objective of in vitro studies is to be able to determine the potential influence of such fields and to isolate them from other type of influence. However, they also have a major disadvantages. Cells or tissue are removed from their natural environment, thereby eliminating the interaction and protection mechanism otherwise available from the donor organism. Moreover, the field used are generally stronger than the field to which the population or worker are exposed. This can result in effects that do not exist with low yield value. It should also be emphasized that a modification that has occurred on a cellular level during tests does not mean that the whole organism would experience the same effect. Now we are talking about the advantages of in vitro studies. Especially important to investigate and identify cellular or molecular working mechanisms. Point 1, you know exactly what you are doing. Point 2, your work can be very specific and detailed. Example, investigation of cell division failure by looking at mitotic spindle apparatus or particular DNA studies that is known as omics. Fast screening, negative in vitro and negative in vivo, relatively inexpensive, often predictive of a real hazard or risk. Example, DNA damage. High throughput screening, example, vetotox tests, omic or specific cell lines, lung or skin epithelial cell, white blood cell and hepatocytes. Now we are talking about the limitation on in vitro studies. Cells are treated outside their normal environment. No surrounding tissue, no blood supply, no normal supply of nutrients. In vivo, exposure cannot easily be mimicked. Metabolization can be stimulated by addition of specific clinical agents. Now we are talking about the models. The first model is ADME experimental model for in vitro. The in vitro metabolic models. In DMPK, drug-like properties commonly refer to respectable absorption, adequate low metabolism, complete elimination from body and minimal toxicological risk. In vitro assay, which play an essential role in screening chemical entities in the discovery stage, have many unique advantages. First, these assays provide a simple, convenient, and fast way to test the potency and drug-like property of chemical entities. Second, in vitro assay require limited amount of test compound, which is appliance since compound supply is limited at the early drug discovery stages, and it is not always feasible to perform preclinical animal studies. Third, in vitro assays are often designed to answer specific questions such as structure-activity relationship, that is SAR. For metabolic stability or DDI potential of drug candidates Answer to these questions may be difficult to obtain in animal studies due to confounding factors. Fourth, human-based in vitro assay could provide a more accurate estimation of human clinical outcomes than could animal tests at the preclinical stage, especially for properties that are known to have species-related difference between preclinical animals and humans, and where a strong in vitro in vivo correlation have already been established in humans. An example of such a property is CYP enzyme induction. It has been shown that preclinical species cannot always predict human CYP induction potential due to species-related difference. However, a good in vitro in vivo correlation has been observed in humans. For instance, a negative induction in human in vitro studies during primary hepatocyte culture seemed to always like to negative induction observed in humans in clinical studies. Therefore, FDA typically waives clinical DDI studies if the drug candidate is tested negative in a human in vitro CYP induction study. The prediction of human efficacy and DMPK toxicology properties from preclinical studies is perhaps the ultimate goal of drug discovery. A common practice in pharmaceutical industry is to establish in vitro in vivo correlation from ADME parameter in multi-species preclinical studies and then find the species that correlate to humans. If the in vitro in vivo correlation is established in an animal species and the in vitro correlation between the animal species and human is also established, then have in vitro information could be used to predict the clinical ADME outcome. On the other hand, if the in vivo correlation of a parameter between animal and human is well understood, then animal in vivo data could be used to predict human outcomes. Example of parameter often used for such prediction include human efficacy, PK properties, example target inhibition, absorption and cleanness, and toxicological endpoint that is QT prolongation. Once the in vitro in vivo or animal to human correlation are established, the estimation, for example, of human clearance from human in vitro clearance data, of human efficacy from xenograft mouse model, or of human QT prolongation risk from dog studies are possible. In general, DMPK parameters are extrapolated from comparison of in vitro data across species and or in vitro in vivo correlation in animals. Notably, in vivo model provide combined effects of permeability, distribution, metabolism and elimination. Thus, they have limitations for such application as to pinpoint a specific factor in an observation. For example, it would be hard to rely only on difference in permeability without considering differences in first part metabolism to evaluate bioavailability among a series of drug candidates. Okay, now we are talking about express enzyme. A primary application of express enzymes is to conduct reaction phenotyping for drug candidates. The FDA DDI guidelines requires conducting cleanness DDI trial if a compound has greater than 25% of its cleanness mediated by a particular pathway. CYP metabolism is the most common clearness pathway for the elimination of drug. Thus, identifying the relative contribution of each CYP isoforms to the total hepatic clearance is an essential task. Additionally, when the CYP metabolism data are combined with the data on other non-metabolic clearance pathways, such as renal or biliary clearance, estimations can be made on the contribution of each CYP isoform to the total body clearance of a drug. A second application of the express enzyme is to identify the involvement of CYPs in metabolic pathway. This assay provides important information on whether the drug candidate is metabolized by the single isoform or by multiple isoform, and furthermore, whether highly polymorphic enzymes such as CYP2D6 and CYP2C19 are the major contribution to its metabolic clearance. It should be noted that while in the most cases hepatic metabolism are performed by the primary drug metabolism CYP, other enzymes could also contribute significantly to the overall metabolism. Example of non-CYP microsomal or cytosolic enzyme often involved in drug metabolism including flavin-containing monooxygenase, that is FMO, and UGT and SULT and AO. Express enzymes are also used for mechanistic application. The example described by Li and colleagues features the use of express enzymes either incubated separately or else in combination with each other. The result illustrated that CYP3A4 was involved in the generation of a metabolite that in turn was responsible for time independence inhibition, that is TDI, of CYP2D6. Additionally, since express CYP systems are concentrated enzymes with high activity toward a specific substrate, they are often used at bioreactors to generate metabolites of interest for further using. Using the generated metabolite that are collected as chromatographic fractions, it is possible to test for pharmacological activity without the need to identify the metabolite structure. In this application, the high activity and selectivity of express enzyme provide a superior alternative to the use of human liver microsome. The potential presence of additional metabolites adds difficulty to liquid chromatographic separation. However, the added expense of using express CYP provide its own challenges. Now, subcellular fraction. Along with express enzyme system, subcellular fraction prepared from drug metabolizing tissue, that is liver and gut, including cytosol, S9 and microsomal fraction are used in drug metabolism studies to address various questions. The cytosol, which is isolated as a supernatant from the S9 fraction containing a group of soluble drug metabolizing enzyme responsible for a specific route of drug metabolism. It is the simplest system of the three. The S9 fraction contains both cytosol and microsomes and represent a nearly complete collection of all drug metabolizing enzymes. However, the presence of so many enzymes can sometimes dilute the activity of the enzyme of interest. The microsomal fraction contains membrane-bound CYP and primary conjugation enzymes such as UGT. These enzymes are responsible for the metabolism of over 90% of marketed drugs. Thus, this is the most frequently utilized enzyme system. Microsomes are the most widely used subcellular fraction for drug metabolism study with the advantage of being inexpensive and easy to handle while containing the major drug metabolism enzyme. One application of microsomal assay is to determine intrinsic cleanness, a useful parameter for facilitating the screening process for stable compound and for establishing an in vitro correlation between animals and humans. And additional features of microsome that they contain relatively concentrated CYP enzyme make them fully for enzyme kinetic study, which provide information on such parameter on the Vmax and Km of a metabolic pathway. The Ki or IC50 for reversible inhibition and the K in ACT and Ki for mechanism-based inhibition. Whole cell system. In this, hepatocytes, other cells and cell lines are considered. Hepatocytes are primary liver cell where drug metabolism occurs. Hepatocyte cells membrane contain various uptake or efflux transporters. Drug could passively diffuse through the hepatocyte membrane or be taken up by transporters such as OATP and sodium taurochlorate NTCP to enhance the intracellular concentration. Once a drug enters hepatocytes, the efflux transporters such as PGP or BCRP and multi-drug resistant associated protein, that is MRP, could pump the drug out to reduce the intracellular drug concentration. The primary cultures of hepatocyte carry enzymes and cofactors at physiological concentration and provide a drug metabolism environment that closely mimic the in vivo condition. Freshly prepared hepatocytes are good model for drug metabolism and transporter studies. However, their application have limitation, including they are not readily available for humans and other higher animals, and a preparation could only be used once. So it is difficult to repeat a study or compare a study between laboratories. These limitations can be overcome with cryopreserved human hepatocyte. Their utility in the in vitro system has provided big advantage in drug discovery. One major application of cultured hepatocyte in for use in CYP induction studies, because enzyme induction may involve gene transcription and translation and therefore may require time to take effect. In fact, primary cultured human hepatocyte have become the gold strand for conducting CYP induction studies. Cell lines such as FA2N4 and HEPA-RG are also used for CYP induction evaluation. However, reports suggest that some of the CYP enzymes such as CYP2B6 may be underrepresented in these cell lines. Hepatocytes culturing for long term has become an emerging area of research in the last decade. While there are some progress, most of the successful examples are limited at academic scale, such as the design of a special format or device for 2D or 3D culturing system, with which especially is required to maintain and perform even a small scale study. None of these systems is ready to be practically used for commercial application, such as industrial research for drug screening and drug development. In vitro transporter models. Transporters play important roles in drug disposition, drug-drug interaction, and drug toxicity. Approximate 400 human transporters can be classified into ATP binding cassette, that is ABC transporter, and solute carrier, that is SLC transporter families. In general, ABC transporters pump the substrate out of cell, whereas SLC transporters take substrate into the cell. Transporter substrate can be endogenous compound or xenobiotics, including some drugs, drug candidates and their metabolites. The key efflux transporter include PGP, BCRP and BSEP, whereas the important uptake transporter are OATP1B1, OATP1B3, OAT1, OAT3, OCT1 and OCT2. Determination of the drug candidate's status, transporter substrate and or inhibit or require performance of a transporter assay using one of the following in vitro experiment model. Immortalized cell lines. Human colon cancer derived CoCA2 cell line is widely used as a model of permeability and efflux transport. This is because CoCA2 cell undergo enterocytic differentiation and become polarized in culture, usually for 21 days, resembling human intestinal epithelium in transporter expressions and tight junction formation. The bidirectional permeability assay using CoCA2 is the most popular method for identification of PGP substrate and inhibition in drug discovery among the various in vitro models. Now we are talking about transferred cell lines. Chinese hamster ovary, that is CHO cell, Marden-Darby canine kidney, that is MDCK cells, human embryonic kidney 293 cells and pig kidney epithelial cell are commonly transfected to overexpress a single transporter. For certain efflux transporter transfected in MDCK cells, co-expression of a corresponding uptake transporter is necessary. The transfection can be transient or permanent. A permanent transfection cell line is usually favored over transient transfection because the former produce more reproducible data. Hepatocytes. The liver play a central role in drug metabolism and disposition through its primary metabolic enzyme, both phase 1 and phase 2, and many transporter, including both uptake transporter expressed on the sinusoidal membrane and express transporters expressed on sinusoidal and canalicular membrane. Therefore, freshly isolated hepatocytes are regularly used to determine hepatic metabolism and clearance metabolites by liver enzyme, as well as to assess hepatic uptake mediated by uptake transporters. In experiment, the uptake into hepatocytes is measured in suspension by a centrifugation method using oil layer tube to separate hepatocyte from incubation medium. There have also been reports that cryopreserved hepatocyte can be used for studying hepatic uptake. Isolated hepatocyte cannot be directly used for efflux assay because the efflux transporters lose their function during the isolation process. However, primary cultures of hepatocytes can restore the function of efflux transporter. Even though the uptake transporter demonstrate decreased function in primary cultures. Now we are talking about the in vitro model for the study of oral delivery. In vitro techniques are useful in modeling the in vivo environment of the GI tract. In models such as simulated gastric fluid and membrane analysis, the modeling of in vivo environment can be done without the use of human cell line, while other models such as transval system require the use of various human cell line. Simulated gastric fluid model how the nanoparticle will react to the fluid of the GI tract and therefore can be used as a preliminary test for the potential behavior of the nanoparticle in the gut after oral delivery. Although the membrane model provides useful information about drug protein transport, it does not take into account the epithelial cell behavior and cannot provide information about the transfection potential for gene delivery system. A more mimetic model for the GI epithelium is the transval system. The system includes a 10 micrometer thick membrane insert that is especially prepared for cell culture and is made out either polyester or polycarbonate. The membrane is available in varying pore size. However, for the purpose of oral delivery studies, the ideal range of pore size is 0.4 to 3 micrometer. Three transwell systems are used to model various cell type and barrier in the gastroepithelium, the CaCO2 monolayer, the CaCO2 and the Regib-B or lymphocyte coculture and CaCO2 and HT29 coculture. In the transwell system, it is possible to monitor nanoparticle transport or gene delivery by using confocal microscopy or flow cytometry. Protein or drug level can be determined the measuring amount in the media of membrane or basal compartment of the transwell. The modeling of the intestinal system has recently been modified to include fluid flow with the development of the gut on a chip micro device. This device is available to better model the human intestine through the use of fluid flow and mechanical stress with the culture of CaCO2 cells. Now we are talking about the simulated gastric fluid. The GI tract presents a unique microenvironment of enzyme and ionic strength which impact both the chemical and colloidal stability of nanoparticles. The typical PBS used to conduct in vitro studies for oral delivery may therefore be inaccurate. Simulated gastric and intestinal fluid provide a medium that is representative of the fluid found in the stomach and intestine and thus are better suited to study and analyze the release profile of controlled release nanoparticle. In the in vitro fluid model, simulated gastric fluid amount at a pH of 1.2 and simulated intestine fluid maximum at a pH of 6.8 without enzymes are prepared according to some standard, oftentimes of USP recommendation of the British Pharmacopoeia, with control release experiment being performed in the test tube. These experiments are typically performed at 37 degrees centigrade and some are done with agitation of 100 rpm to model the in vivo movement of the gastric fluid. Sample of the medium are withdrawn from the test tube at desired interval. The nanoparticles are removed by centrifugation and the amount of drug or protein remaining in the supernatant is analyzed. Drug release study using a membrane. The dialysis membrane model is used for in vitro analysis in experiment that aim to determine peptide and drug transport property. In this technique, nanoparticle dispersion are added to dialysis bag or a dialysis tube and suspended in the desired buffer. For example, in the case of estradiol release from PLGA nanoparticle, ionic strand phosphate buffer at pH 7.4 is used as the buffer. To stimulate GI condition, the membrane system is maintained at 37 degrees centigrade and movement is facilitated by shaking at 100 rpm. The model can be made even more accurate in representing gastric and intestinal fluid with the use of low pH buffer that model the acidic condition of the gut. They analyze the drug release aliquots of the exterior membrane buffer are removed at predetermined time interval for measure of the drug content by models such as high performance liquid chromatography. Now CoCA2 monolayer. The human epithelial colorectal adenocarcinoma cell line, that is CoCA2, is typically used to represent the predominant cell type in the gut. The gut epithelial cell CoCA2 monolayers can either be grown in the monolayer on a single culture well plate or in stress wall, set up to model the transport of drug or passage of nanoparticle across the epithelial layer. With the CoCA2 monolayer grown on a single well cell culture plate, nanoparticle uptake by the cell or transfection of the CoCA2 cell line can be determined through various models. In this transwell model, the CoCA2 monolayer is grown on an insert porous membrane and different cell line can be seeded on the bottom chamber of the plate. For gene delivery study, the model worked by placing the nanoparticle dispersion on the monolayer on the membrane while nourishing the cell beneath the membrane with nanoparticles free medium. Transfection of the cell seeded in the bottom well indicates that the nanoparticle have effectively transported through the CoCA2 monolayer. The CoCA2 monolayer is typically used between 15 and day 25 after initial seeding. The expected TEER, that is transepithelial electrical resistance, is a measurement of the influxes and viability of the monolayer. Typically, a higher TEER value is representative of a more compact cell layer, while a decrease in TEER causing greater cell layer permeability. TEER is an important parameter characterizing the integrity of the cell layer on the tissue, although this system is often used to model intestinal transport. One disadvantage of CoCA2 monolayer is impermeability to hydrophilic or paracellular transport, therefore making it is a better model for clonic tissue and restrict the reliability of technique as a model for the small intestine. CaCO2 and HT29 co-culture. Mucus is significant in limiting transport from the gut fluids to the epithelial cell. The potential trapping of the nanoparticle by mucus lining of the epithelial layer also has an effect on the retention time of the nanoparticle in the system. The co-culture of CaCO2 and the HT29, a human adenocarcinoma cell line, provide a model for the second major cell type in the gut epithelium, the mucus secreting global cell. HT29 cell differentiate into mature goblet cell under the influence of MTX, that is methotrexate, making this cell line important in the mucus layer formation. The culture of CaCO2 and the HT29 MTX is typically optimized by mixing the two cell lines in appropriate ratio and plating them on the porous membrane of the transwell. The viability of the CaCO2 monolayer with the mucus secretion is determined the same way as CaCO2 monolayer alone using TEER. The expected TEER value of the co-culture decrease as the percentage of HT29 MTX increase. The example, TEER range from 0 to 790 from time 0 to 23 days for 25% HT29 MTX, while it ranged from 0 to 710 for 75% HT29 MTX. An alternative way to determine the viability of the CaCO2 and HT29 MTX co-culture is demonstrated by Mahler et al. And involve the mucus layer characterization. The co-culture are seeded on the transwell membrane insert, and after 16 days, a slice of insert is cut out and stained with 0.2% toluene blue for microscopic examination. In one experiment, HT29 subclones are tested for preparation of the most uniform mucus monolayer with some HT29 subclone. The problem of multi-layer cell growth result in only the top layer of HT29 maturing into goblet secreting cell, leaving the CaCO2 and HT29 as multicellular layer belong and limiting the accuracy of the model. However, the HT29DI and HT29E12 clones, both stimulated by MTX, were found to completely mature into globate cell and form a uniform mucus layer with a CaCO2 monolayer beneath it. Different subclone exhibit varying depth of the mucus layer, and therefore they can be used to model different segments of the GI tract. HT29D1 developed into mucus layer of 53 plus minus 52 micrometer, and the HT29MTX E12 result in mucus layer of 142 plus minus 51 micrometer. One limitation of the CaCO2 HT29MTX model is that the active transport of drug is underestimated by the model compared to the accurate result in vivo. Now we are talking about the co-culture for M cell formation. In the intestine, the lymphoid system, such as the Peyer's patch, are separated from the lumen by the follicle-associated epithelium, that is FAE, which contain enterocyte and M cell with few globulate cell. M cell have the capacity of transporting particular matter, including nanoparticles, through transcytosis from the gut contain the underlying mucosal immune system. The initial method for the formation of M cell in the transwell system involved the use of lymphocytes take from the Peyer's patch of mice mixed with the CaCO2 cell line. In the system, the CaCO2 monolayer was allowed formed, and then the isolated lymphocyte were cultured on the basolateral side of the monolayer in transwell plate. The Peyer's patch lymphocyte converted the endothelial cell into M cell in the presence of lymphocytes. The CaCO2 acquired transcytotic activity and were able to transfer fluorescence in thiocyanate conjunctive lattice beard through the filter to the bottom well. In this method, CaCO2 cells are cultured for 14 days, then Regi-B are added to the bottom chamber and the co-cultures are maintained for 4 to 6 days. It is desirable that the epithelial layer lose its microvilli, thus demonstrating the CaCO2 cell live conversion into M cell. However, the loss of microvilli is shown to be non-uniform in the co-culture method, suggesting that some of the epithelial cells remain unconverted. While this non-uniform conversion may accurately be beneficial in representing the distribution of M cell and enterocyte in the GI tract, the M cell formation occur on both the apical and basolateral side of the membrane insert, which is not representative of their presence in the vivo model. Now we are talking about the last method. In this method, a new technique termed gut on a chip provides an in vitro model that mimics the intestine structure and transport properties while sustaining microbial flora. The micro device is fabricated with upper and lower microchannel using PDMS with a 30 micrometer thick porous membrane separating them. The microchannel is treated with an extracellular matrix solution containing red type 1 collagen. and CaCO2 cell are cultured on the ECM coated porous microchannel. The CaCO2 cell experiencing fluid flow in the micro device from monolayer in only 3 days as compared to the 21 days that is taken in the static medium transwell system. The fluid flow during CaCO2 cultures in monolayer allows for the cell to grow in columnar form with microvilli fluids, tight junction and crypt formation that mimic in vivo human intestinal epithelium. Now, we are talking about in vitro cell model for ophthalmic drug development application. In vitro cell-based model in this corneal morphology. The CE is the outermost layer of the eye, which acts as a major barrier of the eye. Since 1960s, many in vitro epithelial models have been established as replacement to Draz tests based on primary and immunized cell line of human and animal organ. Primary model. Most of the primary cell culture models that mimic corneal barrier are constructed from isolated corneal epithelial cell from rabbits. For instance, Cavazo et al. Established a primary rabbit corneal model which resembled the morphology of an intact cornea and was used for study permeability of beta-adrenergic antagonist and levofloxacin. The model tear and the tight barrier were not comparable to that of cornea in vivo. In other study by Chang et al., a primary rabbit cornea epithelial membrane was developed with more distinct barrier property due to air interface culture condition. Model of human primary epithelial cell have also been investigated as toxicology model. Furthermore, human primary corneal epithelial that is HCE cell has been used preparation of tissue sheet to reconstruct the ocular system in severe ocular surface disorder. Some authors such as Ban et al. Were successful in developing a stratified epithelium by culturing human corneal limbal cells on human amniotic membrane, which showed tight barrier junction after 28 days in culture. However, the application of HCE has been limited as alternative in vitro model of corneal barrier due to the low availability of donor cornea. Now we are talking about immortalized model. In this, various models of immortalized animal corneal epithelial cell have been developed from the rabbit, rat and hamster. Model of immortalized rabbit CE cell are more commonly developed and used compared with the other animals. In addition, a rabbit corneal cell line known as Sten's submerged rabbit corneal cell line is widely used, despite showing a fibroblast phenotype, which in turn decreases its value as a valid model for CE. Although these models have helped in reducing the use of animal, the issue related with their non-human origin are still present. In human, to overcome this limitation since early 90s, considerable efforts have been focused on developing in vitro model derived from human cell. Recently, a large amount of research is being done in the field of 3D tissue engineering model. Example of the in vitro human derived CE model that are commercially available as a 3D human cornea equivalents, including EPI ocular, that is MedTech and HCE and Lonza. Another model which shows the in vitro study for ophthalmic drug development applications are conjunctive model, retinal models. In retina, there are two parts, retinal pigment epithelium or retinal capillary endothelium. Next model is immurized model and the other one is ocular disease model and last one is 3D in vitro model of the eye. Now in the last we are talking about the safety tests. So common product safety tests are toxicity LD50 test, eye irritancy risk test, skin irritation, mutagenicity, toxicokinetics and ADME, metabolic toxicity, pyrogen testing, phototoxicity, embryotoxicity, endocrine distributors, ecotoxicity, and the last one is toxicogenomics. Thank you.